Okay, so the next question in discussion is cell death, which is one of your final exam questions and a very important one. Uh, Here we need to discuss about the sequence of events, types of cell death, and their mechanisms. What's interesting is this was one of my final exam questions, so I can tell you from personal experience what you would be expected to know and the kind of questions which they could ask you for this question. So let's begin. Right. So first we start with the definition of cell death. The basic definition of cell death is it is the irreversible loss of a structural and functional integrity of a cell, and the hallmark of cell death is the loss of the nucleus, basically. Um, and there are three ways in which you should be able to describe how the loss of nucleus occurs. Well, it can occur as pyknosis, that is the shrinking of nucleus due to the loss of chromatin, or it can be due to fragmentation of the DNA, which is called karyohexis, or it can be in the form of karyolysis, which is the dissolution and destruction of the nucleus. How it looks like microscopically, we'll see in the next slide. Okay, so here is the normal nucleus. Here is the karyolysis, the dissolution and destruction of the nucleus. This is the pyknosis, where the nucleus is shrinking, and this is the karyohexis, where the nucleus is undergoing fragmentation. This picture is just for comparison how it looks like when it becomes in the form of apoptotic bodies. So this is how it looks like microscopically. The topic cell death is mainly based on the morphology microscopically rather than macroscopic, and how each step and each type of cell death looks like microscopically we'll be discussing in this question. And this is one of the few questions where the microscopic knowledge is more important and very important rather than macroscopy, because in this question you do not really have a lot of macroscopy but microscopy. So yeah. Okay, so the main types of cell death are two, the most important ones, apoptosis and necrosis. But it's also important for you to know some few basic details about necroptosis, pyroptosis, and ferroptosis. So if you name these five, talk in detail about apoptosis and necrosis, and say some basic points about the rest three, you'll be good with the question. Okay, so let's start with apoptosis. Well, what is apoptosis? We all know from our previous studies that it is a genetically programmed regulated cell death, often present physiologically, and it commonly involves single cells, and the membranes remain intact when in the form of apoptotic bodies. Hence, we do not have any inflammation when apoptosis occurs in the body, and when physiologically any cell dies or any tissue dies, we do not see any inflammation. You could briefly mention about the two main pathways of apoptosis, which are intrinsic and extrinsic. Just describe that it is activated by p53 followed by cytochrome C. And in the case of extrinsic, you could talk about TNF alpha a bit, and you could talk about the cytotoxic pathway. But no one expects you to know the whole, all the technical details about the three pathways, like we had in genetics, for example. If you just name them and just give a few lines about what triggers it and what leads the pathway, that's enough for you to know, for them to know that you understand the topic. Okay, but what's important for you to say is the morphology step by step, because without this you cannot, your answer will not be complete. So what is the morphology of apoptosis? And we're talking about microscopy here, not the macro. So the morphology starts with a shrinkage of the cell as well as the nucleus. And how this happens is by the chromatin condensation, which is called pyknosis, shrinkage of the nucleus, followed by blebbing of the cell membrane, followed by nuclear breakdown and collapse, karyohexis and karyolysis, and finally formation of apoptotic bodies surrounded by the cell membrane. And at last, you have lysis of apoptotic bodies, and then they're taken up by the macrophages. The words in which are I've put in color over here need to be remembered. Without these words, your answer is not complete. So please do remember these specific terms. Okay. Some important facts to be remembered about apoptosis: that all pathways, regardless it is intrinsic, extrinsic, or cytotoxic, they all lead to the same result. That is activation of caspases, which will digest the cell. And finally, the cellular debris is packed into the vacuoles known as apoptotic bodies, and then they're eaten up by macrophages, like I mentioned. Well, why do we care about apoptosis? Because it is part of your normal development and maintenance in the case of WBC turnover. So that's physiologic. Then it is also part of some pathological processes. For example, in the case of Alzheimer's, which is a degenerative disease of the brain, Hence, it is important, as well as the atrophy and adaptive changes of some organs. They experience apoptosis, and atrophy usually happens by the process of apoptosis. Hence, we care about apoptosis in pathology. Okay, this is a pictorial representation of how apoptosis happens step by step. So we have a normal cell, followed by a shrinking of the cell and the condensation of chromatin, and that is further followed by blabbing of membrane, sort of like bubbles forming on the border of membrane. Then we have the cellular and nuclear fragmentation, followed by formation of apoptotic bodies in the form of vacuoles. And finally, the apoptotic bodies are taken up by the phagocytes and dissolved. Okay, necrosis, which is a completely pathological pathological cell death, and very important for this question. So this is basically a cell death caused by external insult, in contrast to apoptosis, which could be external or internal, both, and physiological. This is a completely pathological process, and it most commonly involves groups of cells rather than a single cell. And it always causes inflammation. That is the most important point about necrosis, and the point which differentiates it from apoptosis. And why do we have inflammation in the case of necrosis? Well, because the membrane is damaged, and there's leak of the cellular contents into the extracellular matrix, which trigger the immune pathway and starts the process of inflammation. How does necrosis look like morphologically under microscope? Well, it starts with pycnosis, karyolysis, or karyolysis, either of the three. But what's important to mention, other than this, is that in the case of necrosis, the cell swells and not shrinks, and it looks very eosinophilic under the microscope. Whereas in apoptosis, the cell would be shrinking under the microscope. That is what differentiates necrosis from apoptosis. So these three terms, plus a swelling of the cell and eosinophilic nature, are the microscopic morphology of necrosis. Okay, just a pictorial representation of necrosis. This is a normal cell, and it undergoes some form of external insult or injury. You have swelling of the cellular organelles, cellular contents, and then membrane blebbing maybe, but cellular swelling is more important here. And then you have further injury, which makes the cellular injury irreversible. So you start having breakdown of the cell organelles, and which is followed by inflammation and release of cellular contents into the extracellular matrix. Okay, so there are some subtypes of necrosis which you need to know for the exam, starting with the most important one, which is the coagulative necrosis. And it is the most common one, where your cell outline or the tissue outline is preserved, but there's an infarction of the tissue, and it is the infarcted tissue is paler or lighter in color most of the time compared to the original tissue which was there before, like you can see here in the case of heart and kidney. But we also have a sort of red kind of infarct in the case of testes and lungs, and that is because these two organs have dual blood supply, and hence we expect red infarcts instead of white infarcts in the case of testes and lungs. And the nature of the scar tissue after coagulative necrosis is it's harder and paler, basically firm and pale infarct compared to the original tissue. And why is this firm or harder? Is because of the denaturation of proteins present there. And the most common example for white pale infarct would be myocardial infarction and kidney infarction. And the most common examples for red infarct or the red hemorrhagic infarct would be testes and lungs due to the dual blood supply. You see a picture of brain here, but there is that you do not speak about brain over here. You speak about brain in the next slide. Okay, so the next subtype of necrosis is liquefactive necrosis. And why is it liquefactive? Because it is due to the protease enzymes present in certain organs in high quantity, which cause the cells to be dissolved, and that leaves a structureless liquid substance. And which organs are high in proteases? Those are brain and pancreas. Hence, I mentioned when you talk about when I spoke, I do not mention brain over there, but rather mention brain in under liquefactive necrosis. So, which organs experience liquefactive necrosis? You would talk about brain and pancreas because they're high in protease enzymes, and they form a structureless liquid substance when they undergo necrosis. Right. Next, form of subtype of necrosis is gangrene necrosis. It is a sort of mix of coagulative and liquefactive necrosis, not a specific not category. I could put it under. So we could talk about it as a separate subtype. And there are three types of gangrene: dry, wet, and gas gangrene. And usually you see dry and wet gangrene in diabetic patients, or limb ischemia in the case of dry gangrene. And the mummified tissue, it's an important term here to be mentioned under dry gangrene. And this is how a dry gangrenous tissue will look like. And this is how a wet gangrenous tissue will look like. What's the difference between wet and dry gangrene? Well, the difference is that the wet gangrene was basically dry gangrene, which was superimposed by an infection, and it also has pus and foul smell. Hence, it is basically a dry gangrene which became wet gangrene because of the presence of infection, and mainly happens in decubitus, basically bed sores in hospitalized patients or diabetic patients. Last is gas gangrene due to the anaerobic bacteria, most commonly Clostridium perfringens. It is a gas-producing bacteria, and it is a very bad situation. It is a life-threatening infection, basically. This is the picture of a decubitus or a wet gangrene, and this is a picture of the wet gangrene on the foot of a diabetic patient. Okay, the next category of the necrosis subtype is caseous necrosis. It is a subcategory of coagulative necrosis, basically. So it actually comes under coagulative necrosis, but due to its nature of morphology, we give it a separate subclass sometimes. But if you say this, the examiner will understand that you know step. What is its morphology? Its morphology is it's a cottage cheese-like texture, soft, which can be spread with a knife, for example, and it's mainly seen in tuberculosis. Next is fat or bulbous necrosis, which is a result of saponification of pancreatic or peripancreatic fat. This is different from the liquefactive necrosis, which we saw here. The liquefactive necrosis of pancreas is of the pancreatic tissue, whereas the fat or bulbous necrosis is of the peripancreatic and the pancreatic fat. So this is something important to note. And why does it happen? And what exactly happens here? Saponification is basically formation of soap, and it happens when the free lipases deliberate free fatty acids that combine with calcium, and they form a soap-like substance. The morphology is chalky white appearance. We'll see how it looks. So this is enough for the fat necrosis. The last subtype of necrosis is fibrinoid necrosis, most commonly seen in blood vessel walls due to damage to the blood vessel walls. And it results in deposition of fibrin and immune complex around the vessels, and mostly seen in malignant hypertension and vasculitis. Okay, so this is how fat necrosis looks like. Notice the white chalky morphology around the pancreas, and this is how caseous necrosis looks like in a tuberculosis lung. If you spread it with a knife, it will spread like a cheese. So that's why we call it cottage cheese-like appearance. Yeah. So this is something to remember. Okay. Other types of cell death, which I mentioned when I started the presentation, are necroptosis, ferroptosis, and ferroptosis. You do not need to know too much about it. Just one line and one example should be enough, and your examiner will be happy. At least when I had my exam and I spoke about this, my examiner just asked me to say one line and one example for each. So I think this should be enough for the examiner, because they are not as common as apoptosis and necrosis. So what exactly is necroptosis? Necroptosis is basically something between necrosis and apoptosis, like a hybrid, in which the cell commits suicide when regular apoptosis is inhibited by either viral or tumor mechanisms. It can be considered physiological necrosis in some cases. And what's important to note here, it causes inflammation. And the most important example for necroptosis can be viral hepatitis. And the last two types of cell death are pyroptosis and ferroptosis. Pyroptosis is caused due to intracellular pathogen infections, for example, Shigella, and it is highly inflammatory, releasing IL-1. For the last one is ferroptosis. It is an iron-dependent cell death, and it can actually be useful, for example, in chemotherapy, and it is something still being developed, the concept of ferroptosis. So we do not have much information about it currently.